Hello, welcome to a video on Rho-dependent termination. I'm Stephen Harvey, and today I'll be explaining some of Jeffrey Roberts' 1969 experiments regarding aspects of Rho-dependent transcription termination, specifically regarding figures 6.44 and 6.45 from Robert Weaver's Molecular Biology, 5th edition. First, we'll address Rho's ability to produce shorter transcripts by either transcription termination or RNase activity. And then we'll see evidence that illustrates whether Rho releases transcripts from the DNA template. This video has been produced for MCDB 427, which is the University of Michigan's Molecular Biology course. Go Blue! As background information, Jeffrey Roberts had discovered the protein Rho and its apparent action in causing termination of transcription. To orient ourselves, let's briefly review the steps of transcription, which you should be very, very familiar with. First, initiation. Here, RNA polymerase, henceforth referred to as RNAP, searches for a promoter. The double-stranded DNA is in blue, and RNAP is in purple. Obviously, these drawings are not to scale and do not depict the actual structures of these molecules. Next, a closed promoter complex is formed when RNAP is bound loosely to the DNA. Lastly, RNAP is bound tightly to the DNA promoter by melting a local region and forming the open promoter complex. Second, elongation by the core polymerase. Here, the transcript is elongated as nucleotides are added to a growing RNA chain. We introduce RNA in red with the NTPs seen as these little squiggles. Lastly, in the focus of our video, we have termination. In prokaryotes, this may occur by intrinsic terminators, or without proteins other than RNAP. This is known as Rho-dependent termination. This depends on the actual sequence of the nucleotides themselves. Additionally, with the help of an auxiliary factor called Rho, we have Rho-dependent termination. Throughout the video, Rho will be drawn as this little yellow molecule, without the cape. Next, let's take a brief step backwards at this point and explore the path that Roberts took from discovering Rho to determining its function in Rho-dependent termination. Roberts found that DNA transcription in the presence of Rho resulted in a depression in the level of transcription, seen as reduced RNA. One explanation for this would be that Rho causes termination events. When termination occurs, RNAP and our nascent transcript separate from the DNA template. RNAP must then reinitiate again with the DNA, which is a time-consuming event in this cyclical process. Thus, net transcription decreases. Initiation is our time-limiting factor. This was a great idea. Roberts was brilliant, but could he prove it? Roberts knew that the addition of Rho causes production of shorter transcripts. He suspected that this was the result of termination. But what alternative mechanisms could result in shorter transcripts? Pause this video, think about it. Write your answers on a $20 bill and mail it to the University of Michigan MCDB department. Now that you're back, let's see how you did. If you said that Rho could act as an RNase and simply chop up the transcripts into shorter pieces, then you are correct. If you said something else, then it may be a good idea that you're watching this video and learning. To rule out Rho acting as an RNase, Roberts followed the following method. First, Roberts took lambda DNA, added E. coli RNAP, and added NTPs. This is what's needed for transcription. But to assay his results, he added tritium-labeled UTPs to make his RNA transcripts radio-labeled. With his components combined, which would actually not be visible in the beaker, transcription led to long tritium-labeled transcripts. How did Roberts know the length? He used ultracentrifugation to separate them by size. Let's quickly discuss this to ensure we're on the same page. By now, you're assuredly familiar with the separation of RNA by electrophoresis. However, Roberts used ultracentrifugation. A sample is spun at high speeds. in a tube, allowing the constituents to differentially sediment according to their physical properties, or in our case, according to RNA mass, which relates to length. In the test tube, at the bottom, you'll see increased mass, and at the top, you'll see RNA of lower mass. Collecting fractions, portions of sedimented RNA, from the bottom of the centrifuge tube, Roberts separated the RNA. Fractions in the tube will be counted like this: 1 at the bottom, then 2, 3, 4, and 5 at the top. The number of fractions would vary in a real experiment. This is important, so understand this next concept. Longer RNA sedimented on the bottom of the tube. Thus, earlier fractions contain longer RNA. Logically then, higher fractions, labeled 4 and 5 in this example, will then have increasingly smaller RNA transcripts. Remember that these transcripts contain radiolabeled UTPs, so for detection, what assay would Roberts use? A generic answer would be autoradiography. More specifically, he likely used a scintillation counter. Now that we understand the methods, let's look at some results. Roberts plotted this all on a graph seen here. On the graph, for the x-axis, we see fraction numbers, with smaller fractions correlating with larger RNAs, and larger fraction numbers correlating with smaller RNAs. On the y-axis, we see CPM, which stands for counts per million. This likely refers to a measure used by scintillation counting. This can be used as a measure of the amount of RNA within a certain fraction. In the graph in blue, we had the transcripts that were made. This acts as a controlled distribution of RNA made in the absence of Rho, which we can compare later to our experimental group. We don't know the actual sizes of the RNA, however, just a relative distribution of the fractions. If you remember the control methods, the next portion should be simple. In addition to the DNA, RNAP, and NTPs, Roberts added 3 new items. First, he took his tritium-labeled RNA from the control reaction and added it into the beaker. Additionally, he added labeled UTPs, labeled in a different manner than his previous RNA. Lastly, he added Rho. The 14 carbon-labeled transcripts, along with the former tritium-labeled RNA, were then ultracentrifuged and fractioned off, quantified by a form of radiography. Again, the colors are for understanding and do not actually represent the appearance of fractioned RNA. Here, the fractions were assayed and graphed. Clearly, the original tritium-labeled RNA, seen in blue, was unaffected by Rho. However, the carbon-14 RNA, seen in red, labeled with the additional y-axis, shows a different distribution. There is relatively little of this RNA in the lower fractions, and a great deal of the RNA in the higher fractions. What does this tell us? Pause your video and make some scientific conclusions for yourself. Welcome back. Let's discuss what we can conclude. First, the transcripts made in the presence of Rho, seen in red, are generally shorter or found in larger fractions than the transcripts made not in the presence of Rho. Also, because the tritium-labeled RNA were not shortened when incubated with Rho, we can rule out Rho acting as an RNase. Therefore, this supports Roberts' suspicions. Case closed on figure 6.44. Roberts illustrated that Rho shortens transcripts by causing termination. Another win for science. Moving along to figure 6.45 regarding Rho releasing transcripts from the DNA template. How can this be tested? Here's a hint. It's similar to what we've just learned. Pause your video, petition your local elected officials to increase public university science research funding, write down your ideas, and then press play. Welcome back. To determine whether or not Rho affects release of transcripts from the DNA template, Roberts compared the sedimentation of RNA made in the presence of Rho. What does this tell us? Well, if termination occurs with Rho, which it should, RNA will be released from the DNA and they will sediment separately. However, with Rho, the RNA will still be attached to our template and they will co-sediment. The methods. Roberts added his labeled DNA, which was labeled with any label besides tritium, RNAP, NTPs, and tritium-labeled UTPs all together. He ran the experiments twice, once without Rho and once with Rho. After transcription, the product was ultracentrifugated to see if association with the DNA template existed. Roberts fractioned off the RNA and DNA and then performed some autoradiography and graphed the results. Lo and behold, graph 6.45a on top. With similar axis to 6.44 with our fractions on the bottom and our counts per million on the sides, DNA is labeled in blue, seen here, and RNA is labeled in red, seen here. We can clearly see that the DNA and RNA appeared to co-sediment as their peaks align. This serves as our control minus Rho trial. Next, after the same procedure, except with transcription occurring in the presence of Rho, we have different results. Here the peaks of RNA and DNA are not found in the same fractions. RNA sedimented into fractions corresponding to smaller RNA fragments, while the DNA appeared to sediment where we would expect free DNA to sediment. From this, what can we conclude from the figure? Well, first, in the presence of Rho, RNA release from the DNA template increased. This can be seen by the differing RNA and DNA peaks, illustrating independent sedimentation of RNA and DNA. Next, in the presence of Rho, the distribution of the RNA shifted. It went from graph A here to graph B here, probably from about fraction 8 all the way up to maybe fraction 17. Lastly, there was a shift in the fractions in which DNA is seen from graphs A, here, perhaps around fraction 9, to here, maybe around fraction 12, comparing the RNA-associated DNA to the free DNA. These are not the only conclusions from the figures, but it's a great start. Feel free to keep brainstorming to test your understanding. To summarize what we learned, Rho causes shortened transcripts by initiating termination. Also, Rho does cause release of transcripts from the DNA template. That's the end of the video. I hope you enjoyed yourself and that you learned a little. This is Stephen Harvey, signing off.